Yeah, blocks on its own.
Does the tree open?
Let me just put the code up front. All right. Well, thank you for the wonderful introduction, Dr. Mason. If you didn't know who I am, Brandon Farr, Bob Ray, if you're into that, if you're lazy. So, by a show of hands, I asked this before the presentation, but I want to ask again, with artifacts. How many of you have received your flu shot this season? How many of you have received a flu shot ever? So what has been your experience with that? It doesn't really work out as well. It seems like every time you get a flu shot, you end up getting the flu, right? That's what it seems like. The validity of that claim is a little bit dubious, and I'll get into why that is later in the presentation, but you understand that we've all had the flu. It's horrible. It puts you out of commission for a while, costs quite a bit of money. It's just not worth it. And so when going into the Dr. Mason's lab, I figured this would be a prime opportunity to maybe improve the seasonal flu vaccine. And so without further ado, I present to you the development of a plant-expressed M2E-based universal influenza vaccine. So let's start off some, what's the burden? So what is the actual impact of the flu? And don't worry about the burden, it goes away really quickly. In the United States, from 2010 to 2013, there were roughly 115 to 600,000 hospitalizations in the United States, roughly 19,000 to 85 or 95,000 ICU admissions, and roughly 5,000 to 27,000 deaths in hospitals because of the flu. This number can reach upwards of 49,000 deaths per year in non-pandemic years. This is not like H1N1, like swine flu. This can just happen, just kind of like in a year that's a particularly bad flu. For context, what does 49,000 people look like? Well, that's about the population of the ASU Tempe campus. A little bit under it, actually. That's a lot of people. It'd be awful if that number of people actually, if ASU Tempe just disappeared, right? The economic burden is even worse. The 2015 flu season alone cost the United States economy $5.79 billion. For reference, that is 9.5 times ASU's endowment. That is an enormous amount of money. And that is just the 2015 flu season. Can you guys remember the 2015 flu season? I can't. I don't know what the strain was then. A future pandemic flu, however, could cost $71.3 billion to upwards of $150 billion. Just the American economy. That has nothing to do with the global economy. There's an obvious need for improvement in flu vaccines. How are we going to do it? Well, obviously the CDC yeah.
So can we go back a slide real quick?
Sure.
Those numbers, the hospitalizations and ICU admissions, is that the 2010 numbers, then the 2013 numbers, or are those estimates?
Those are ranges. So those are like estimates. Yeah.
Okay.
And these are, of course, it should be considered that these are all from only cases reported to hospitals. There's by a factor of anywhere from 2 to 5 times more is what the research is predicting that actually exists in the United States. And at the end of the presentation, if you want, I can go into more of how effective the flu vaccine is, and you'll just see how low these numbers are compared to the real thing. Anyone else? All right, cool. So current vaccines are not effective enough. This is something that we all know and have experienced, despite the fact that we all should be getting the flu vaccine. A meta-analysis of 5,700 articles and 31 eligible clinical studies found that TIV vaccines, which essentially are composed of inactivated flu virus, 3 different strains, usually 2 A strains and 1 strain of influenza B. They work in of 8 of 12 seasons. It works about effective in 8 seasons. That doesn't necessarily mean it cures the flu. You're going to get the flu. You're not going to get the vaccine. You're not going to get sick. This is just going to be effective, which they measure as either protecting completely or alleviating some of the symptoms, which is generally what we get when we have the flu vaccine. Obviously, we're not going to get the full-blown flu. We don't end up dying, like in the Spanish flu, but we have lesser symptoms, and that's what they're talking about. And 67% of the time for TIV vaccines, you're going to get lesser symptoms or possibly protection from that year's flu.
Brendan, is it known how many of those 50,000 that are estimated to die this year, or the ones that you actually have numbers for, were vaccinated?
Oh, I don't have those numbers on these, but I have found those numbers, and they are fairly lower. I mean, generally, if you get the flu vaccine, you're not going to have a fatality or mortality.
So most people that get hospitalized and die are the ones that did not get the vaccine.
And generally in these trends, or in the previous slide in these trends, these deaths are generally people age 65 or older who can't really get the vaccine for considerations of compromisation and stuff like that. The 65 and up age group is generally very susceptible to the flu. It's just what the research has found. So then for the live attenuated influenza vaccine, these are vaccines that are not, these are viruses that are not inactivated, but instead grown in selective media or different conditions to make them less virulent, less harmful. And those are effective in 75% of seasons. But again, that doesn't mean you're getting protection. You're just getting alleviation of some of the symptoms, and you're probably not gonna die. This is the point that I wanted to bring up. This is the major take home. Seasonal influenza vaccines, these are seasonal influenza vaccines, show efficacy in 6 of 17 analyses. And what's the difference between this and these? These generally are like, you can get maybe like prophylactic vaccines, but also more importantly, this is measuring very specifically how effective were the strains chosen in the vaccine against that season's flu. So like this season was H3N2. For instance, this season, it was about 25% effective. But against other strains of the flu, it was like 75% effective. So we are really good, even if we are really good at choosing, which we aren't, you're gonna end up with a 35% protection rate anyways, in 6 of 17 analyses of this H12 season, of this 12 season period. Suffice to say, I mean, these are a bunch of numbers I'm throwing at you, a ton of impacts. Flu vaccines aren't effective enough. The way we're doing it isn't effective enough. And we are, even if we get really good at choosing them, it's not effective enough. So that's why we should ask, like, why is it so ineffective? Like, we have all this scientific knowledge. We have a giant research lab just here, and there are hundreds of universities around the United States and the world. Why is this so ineffective? Well, it's the flu structure. The flu is really, really hard to design vaccines for. Why, though? Well, a little bit of background on the flu, for example, just for your consideration. The flu is a Baltimore Group 5 virus, and what that means is that it's negative sense single-stranded RNA virus, which, in order for it to be transcribed, it first needs to go from negative sense to positive sense, and then it uses other machinery to get transcribed again and again. Baltimore Group 5 viruses mutate at one of the highest rates of all Baltimore classes, which tells you immediately this isn't good. It's going to be hard to vaccinate against. Just based on the kind of virus it is, it's going to mutate like a lot, all right? But where is it mutating? Like, what is it mutating? What do we make vaccines out of? Well, we make vaccines out of what the stuff the immune system can see, and that's what's on the outside of the flu. There are 2 major proteins on the outside of the flu. Hemagglutinin is the first one. It's the H in, like, you know, the HXNX notation, H1N2, etc., H1N7. Hemagglutinin is a homotrimeric type 1 integral membrane glycoprotein on the surface of the flu virus. What does that mean? It has 3 copies of itself, and they're all the same, you know, protein. It's not, you know, complexing with something else. And the type 1 integral membrane protein essentially just means that, like, it has a cleavable N-terminal sequence, has a transmembrane sequence here to the, you know, the carboxyl terminus. It essentially, for those who are less familiar with biology, it just goes through the flu's membrane. So it's not just kind of sticking up, it's also embedded in the membrane. So it works, this is like one of the major targets of most flu vaccines because it is really what's responsible largely for entry. What it does is it binds to sialic acid containing receptors, and it essentially like helps it kind of like get attached to the cell that's trying to infect. It actually doesn't have very high affinity for sialic acid, but there's a ton of this hemagglutinin just like on the cell surface, and that's why we're trying to target it, because it's like the biggest marker. And if we can neutralize it, then it can't enter. So there's another one, there's the N, neuraminidase, it's the N. It's also homotetrameric, which means it's step 4, it's only step 3 like the last one. It's type 2 integral membrane protein, which has a non-cleavable hydrophobic transmembrane domain, which is closer to the N-terminus. And it's got like a combined like signal and anchor sequence, which makes it a little bit harder to kind of like deal with. So it removes sialic acid residues, unlike hemagglutinin. That kind of seems like a contradiction, right? You need sialic acid to get into cells, but this gets rid of it. Why would it do that? Well, it needs to do that because if it doesn't, hemagglutinin is just going to stick to the cell, and the virus won't be able to leave the cell after it replicates all this stuff and gets out. It won't be able to get out because it'll be stuck to the sialic acid. So it's important to have a balance of H and N for the flu. Without N, it can't get out. Without H or N, it can't get in or out. So really, if you can find a way to neutralize H, that would be preferable. But if you neutralize N, maybe it would like help a little bit more. Like if you could like enhance an HA-based vaccine. But really, HA is what you want to be targeting with vaccines, and I can argue that to the death. So HA and NA mutate very frequently because evolution works like this, right? If you have something that's giving you selective pressure against you, if there's something selecting against a trait, it will be selected against. It will kind of be reduced in the population. So how does the thing survive? How does the virus survive? Well, it mutates so that it can't be recognized by the immune system anymore. HA and NA have an enormous rate of mutation. Really, really high. I don't have the exact frequency, but it's enormous. I have a paper, I can look it up if it's really important, but I think it's suffice to say, we all understand that it mutates very quickly. For antigenic drift, which is just small mutations in HA or NA as being transcribed, as I mentioned, it has to go through a lot of steps to get made. Anything in there that has a mutation could be antigenic drift. It drifts, it changes kind of like whether it's H1 or N3, H1, H3, H7, etc. You also have antigenic shift, which is a much bigger problem, in my opinion, but again, this is a defense. I will defend my opinion, if you'd like me to. Antigenic shift involves having, the way that I understand it, is you have two different strains of the virus infecting the same cell. And so you can have, if you have H1N1 and H7N7 in the same cell, when the virus buds, you can get H7N1 or H1N7. And so you get a whole different strain of the flu. It's a really big problem, especially when you consider there are 18 possible types of hemagglutinin and 11 types of neuraminidase. That's 498 possible combinations of HMN. Currently, H1 and H3 are the main subtypes affecting humans, but there is really no real reason why we couldn't possibly, maybe at some point in the future, get H2, H6, 7, 8, 9, 10 through 18, etc., etc. It's important that we have a better target, because with that level of variation, it would be best just to not really bother with this for now. I mean, I'm not saying we shouldn't look into better HA vaccines, but there has to be something better, right? There aren't just two proteins on the flu, just entry and getting out. There are more. And what is more? N2. It is the third protein on the flu surface, and it's expressed at far lower levels than HA or NA. In fact, it's really, really low. It's very low. It's not going to neutralize anything. You have antibodies against it. You're not going to neutralize the flu. It's just too low. It's expressed at high levels in infected cells, so we'll get into more of that in a little while. M2 is a homotetrameric type 3 membrane protein. There are a lot of types of membrane proteins. And actually, I couldn't find out how type 3 is different from type 1 and 2. I looked, and I'm feeling bad because I know we learned it in MBB 347. I'm sorry, Dr. Mason. But I can't describe to you why that's different than type 1 and 2. I'd be happy to look into it later, though. Thank you. And it plays a big role in viral encoding. So what is it, though? How does it do that? Well, here's M2E, but I want you to see this kind of structure, okay? This is going to work as a proton channel. It's going to bring protons inside the flu capsid and help acidify the inside of the flu to weaken the bonds holding it together to kind of just out inside of the endosome, which is a vesicle that cells use to uptake exogenous particles. That will allow it to uncode, get all of its machinery out, get all its DNA out, and enter the cell and start hijacking it. So this is pretty important. But as you can see, freaking 90% of it is not targetable. M2 is largely inside of the membrane. So what do? Well, you go after what's on the outside, right? Fun fact, what's on the outside hasn't really changed since 1933. And a few amino acids, as you can see, this underline N and I, those ones have shown some rates of mutation, especially in avian flu. But aside from those three, they don't really mutate, and the amino acids those mutate to are pretty conserved, which is good. So you get 1 or 2 versions of M2E that we can target using a vaccine. It's great, right? It's not going to mutate. If we make a vaccine against it, it will continue to work for most strains of the flu. Also, I should note that the first 9 of 10 amino acids, as you can see, really don't mutate at all. That happens to be the largest T cell epitope that this kind of thing elicits. Anti-M2E antibodies that anti-M2E T cell receptors generally are going to feature that SLL, TEV, TP epitope. And so because that's not changing, it hasn't really changed at all, we'll be sure to get some T cell killing of infected cells. And again, I'll get more into that later. So, on its own, it's really poorly immunogenic. Okay, it's super poorly immunogenic. You really don't get very high antibody titers if you would vaccinate with it. But previous research has overcome this by conjugating it or fusing it to different adjuvants and platforms, including keyhole lipid, H. Let's see, hepatitis B core antigen. You can get some kind of virus-like particles with that. You can get OMPC, which is another adjuvant that they've used in previous studies, but none of these really had super high vaccine titers. The KLH had 1,600 at first shot and 40,000 after booster shots. HBC, you got 4 times 10 to the 4th anti-M2E titers, but you get way higher anti-HPC titers to the magnitude of 10 to the 6th. And for OMPC, you get in the ballpark of like about 1,000. So these aren't super high antibody titers. I mean, it's better than nothing, but there are definitely platforms and methods in our lab that we've used to elicit larger antibody responses, especially in some of the unpublished work of Dr. Dinas that I've seen, Dr. Andy. So yeah, these are good, but we can do better. And these have shown protection against the flu, but we can do better. How are we going to do better? Well, we're going to fuse it to a platform, and we're not really going to matter. It's not going to matter as much if it's tetrameric or not. It's kind of shown that we don't need it to be tetrameric, even though it's native conformation. We've got like 4 copies of it, just kind of flinging like little noodles on M2. But we don't really need to do it as much. One study showed that it didn't really result in differences in anti-M2 antibody generation. So we're going to do this in plants. And don't worry, I will give you the punchline. I'll tell you what it's going to be. But first I need to tell you we're going to do the plants. Why are we doing this in plants? Well, simply put, Zach actually has a vaccine. He can't take the flu vaccine because there are certain antigens in the vaccine through the method of production. I figure that he's probably not allergic to plants. That would probably kill him if he were. He would never be able to eat salad or any plant antigens. So there's that. It's good. It's viable, we can do it. I mean, the fact that our labs exist, Dr. Moore and Dr. Mason's lab exists, means that we can do this. It's efficient, it can be done really well. We can produce it at very high titers. We can produce whatever we really want at very high concentrations, as long as the method of the vector design is good and what the elements are in the vector are well designed. And it's cost effective. Growing plants is a whole lot cheaper than growing human cell lines. Human cell lines and animal cell lines are super picky. And chicken eggs are generally more expensive than plant seeds, and water and soil and sunlight. So it's a lot cheaper. AA can be also faster. That's one other thing I should mention. Literally what you're going to see, we designed it, and that process we could have done within like a week. We can put it into plants, get it in 5 days, extract it, and purify it, and we have a vaccine in high concentrations done cheaply in like a week and a half, which is absurd. Currently in the CDC, it takes months to get the necessary amount. You usually do it in chicken eggs. It takes months to get the vaccine. So this is good, especially for like new outbreaks. If we find out that M2 mutates, we could just make another one like a week and a half and have it ready. So that's why I'm choosing to do it in plants. So now the platform that I've been talking about, this whole thing, how we're going to get high antibody titers, how we're going to actually make the vaccine. What are you talking about? Give me a background for like 10 minutes. What did you actually do for your thesis? So we are using a recombinant immune complex. Immune complexes in general, and recombinant immune complexes are incredibly immunogenic. They can be expressed in plants. We've shown that, and there are several other authors have shown that. They have been evaluated as a potential vaccine candidate for HIV, and they are investigated as an adjuvant to improve the TB vaccine, as well as other potential. There are other RICs out there that have been made, but these are the 2 I'm choosing to kind of show, just because HIV and TB have big impacts. This makes them a prime candidate as an adjuvant to M2E. Structurally, there's 2 parts. You have an antibody, which, for those of you who are unfamiliar, looks kind of like a Y. You've got a heavy chain, and then you have one light chain. So the arms of the Y have kind of like this. And you'll see a picture on the next slide. And then an antigen fused to the antibody in an epitope tag that the antibody can recognize. And I'll show you how that works. So this right here, this is the monomer. Well, I mean, technically this is monomer. But like this is like a single unit of our RIC, okay? This is our antibody. We have our heavy chain here in the blue. We have our light chain here in the red. We fuse antigen to the carboxyl terminus of the antibody, and then we put an epitope tag. It's just something that the antibody can recognize. This thing right here can recognize this purple bit right here, all right? And we fuse that to the antigen, after the antigen generally. And so then you end up getting these enormous complexes as you have one head binding to the purple head of another antibody. It just keeps growing and growing and growing. And that is fantastically immunogenic. And I can describe why if you'd like to know. Okay, cool.
No, by all means.
So,
you back.
essentially, this part right here is considered the FC gamma portion. It's the constant region, it's the conserved domain of the heavy chain. Your immune system is really good at recognizing this. That's actually the largest part of how immune system how antibody-independent cytotoxic cell killing kind of works. It recognizes the heavy chain, the concentration of the heavy chain of an antibody bound to an antigen, which, say, for M2E or the flu, it's going to be bound right here, right? It'll be sticking up, kind of like a red flag. And so you have macrophages that will recognize this using Fc gamma R receptors. This is an immunoglobulin G, so gamma, Greek, G, we're super cool, Greek. And so you have the immunoglobulin G. It recognizes this portion, and that lets it do a few things. It can traffic an antibody containing an antigen to a lymph node, which will help proliferate a response to that specific antigen. It can also induce cytotoxic cell killing. When the Fc gamma receptor binds to this, it phosphorylates ITAMs inside of the cell, resulting in some transcriptional differences and allowing it to actually go and kill the cell that has this antibody on it. And also, the other thing that this can do is it can activate the innate immune system. C1q can bind to, well, this part again. This is a really important part. And what that does is C1q cleaves C1R, which is a serine protease, which also cleaves C1s, which is another serine protease, which creates the C1 convertase complex. It does a whole bunch of steps, and eventually you get a pore. I have the entire complex cascade written down on a slide if you guys want to see it, but I feel like C1, C2, C3, C4, we could be here all day. There's a lot of complement that's involved. But so that's the two major ways it does it. You've got the Fc gamma receptor being used to traffic it to the lymph node. You've got Fc gamma mediated cell killing, and you also have complement mediated cell killing. So this is great. But the thing I want you to focus on is the fact that this is used to traffic antibody and antigen complexes to the lymph node. That's where the antibodies that you're going to make are getting made. That's how vaccines work. You traffic antigen to the lymph node and it makes a bunch of, it proliferates a B-cell T cell response. I can get into that, but I feel like you probably don't want to know that. Yeah, at least. Maybe later. So yeah, is there any questions so far? I want to stop and just ask, because the next part's kind of like, we did this. I want to make sure you guys understand how this works, at least enough. Any questions at all? Okay, cool. So, the M2E RSC to date, there's never been an M2E RSC. No one's ever made it. Not in plants, not in animals, not in humans, hopefully not in humans. That'd be really weird. We should probably get the FDA on the line for that. Past research has indicated, however, that tandem repeats of M2E fused to platforms perform better than just single M2E repeats, so we did that. We actually did a dimer of it. We were planning, initially in the very beginning of the thesis, we had planned to do a tetramer, because we figured maybe it would kind of look like a tetramer, with the human consensus sequence of M2E, which is what I showed you, and then also the avian mutated sequence that I had indicated previously, and repeating like this. But the company that synthesizes the gene blocks deemed it a little bit too repeat heavy, and there were some problems with synthesis, so we just ended up going with the 2x M2E with a human consensus sequence, with a few codons alternated, just to kind of minimize the potential for it to kind of like go like that, and also have problems. So yeah, that'll be a future correction. If we find out this works, we'll find a way to get this, this heterotetramer of M2E going. So there are 5 major steps to making this RIC, and this is a fun one. There's a lot of pictures. So there are a lot of pictures at some point soon. Step 1, we designed the sequence. So we designed this consensus sequence. I went through like a bunch of papers, like a lot. Like I think I went through like 20 or 30 papers just to make sure that this was actually a human consensus sequence. Because a problem in the literature for some of this like vaccine research is that like, oh, this is the consensus. No, this is the consensus. So I wanted to make sure that this is actually the consensus sequence of human M2E. And thankfully, there was not really that much variation. Actually, there wasn't any variation in the human consensus that I could find. But again, there might be one paper out there that I missed. And we codon optimized it using a codon table to express it in benzamide. And the reason we did that is that different organisms have different rates of recognizing the same codons. Codons are just like a sequence of 3 nucleotides. And so like AUG is the start codon, it's pretty conserved. But then say, for example, another codon like ACG might not be recognized as well by humans or mice as it is in plants. So you gotta make sure you use the right language for the plant. We designed the homologous M2E dimer, we used a glycine serine linker. The reason we did that is because essentially glycine is a small amino acid and serine is kind of, has a hydroxyl group. It's a polar residue. And so that will allow it to not really get too clunky with itself. The small size of the glycine combined with the polarity of the serine really helps it avoid like sticking too much to things we don't want it to. And also, if we use a bigger linker, like bigger amino acids, that can end up having some unwanted interactions with our protein, I can get into that more in the closed defense or afterwards, but I feel like I don't want to waste too much time on that. And we added some restriction sites to put it into an existing RIC vector that was already made for a different project. So step 2 is just building the construct. I promise you guys pictures. We PCR amplified it, we digested it, we gene cleaned it, we ligated it. All that means is we took the gene block that we ordered, we blew it up to really like a huge number of copies of M2E. We then took the ends and cleaved them using restriction enzymes, which essentially cut specific sequences and generate sticky overhanging ends that can be used to put into something that has the complementary end, like this. Then we gene cleaned it, which means we ran it on a gel and we got it out of the gel. And then we ligated it into this the vector plasmid, which already existed. This was an existing RSC plasmid with these specific restriction sites. And so we could put it in here by restricting this as well with the same enzymes. We removed the GD antigen, which was for, I believe, HSV, and we replaced it with M2E. That's essentially how it worked. And this vector plasmid was prepared by Dr. Mason, incorporating like lots of elements from like Andy's research. So, I mean, there's definitely credit to me too. This plasmid worked wonderfully. I definitely could not have designed it any better, or even half as good. So this third step is we transform bacteria with it. We transform E. coli with it to kind of hold on to it. It can amplify it. It can generally, we use this as an amplification kind of storage thing instead of storing it on its own. We also have it in a buffer, the plasmid itself in a buffer in a freezer. But having E. coli hold on to it lets us kind of store it for longer and also amplify it. So we transform chemically competent DH5 alpha cells with electricity. So we made these cells very permeable to just stuff on the outside, and we zap in the plasmid. It's super cool. It sounds kind of scary in the lab. I get scared sometimes from it. And so this is a gel of 10 colonies of our E. coli cells, and we use M2E specific primers in the PCR. Which all it means is that for polymerase chain reaction, you have to use primers that bind to specific sites so that it will actually get amplified to 1 million copies or so, actually way more than that. And if you get amplification, you'll get a band on this gel. This is like DNA, essentially. We're running DNA through a gel that has different sizes of pores that can let sizes of DNA get through at different speeds. And then we transform this in, we did the same thing with A tumefactions, which is agrobacterium tumefactions. That's what we're going to use to get this into the plants. It took a few tries. It was, this is like the final gel. There were like 4 other gels. We did this like 4 times. And so we used 2, just to make sure, because this is what we're going to put in the plants, we used 2 different sets of primers. The Rep R, Rep F used the gem-viral replication proteins. As like kind of like a site, just to make sure that like this PCR is good. And then also we use the epitope tag and the extensive 3IR terminator, which are elements of the plasmid, just to make sure that it's actually there. 68H is the epitope tag. It's the purple thing that I showed you guys on the previous slide. So yeah, this is live footage. So step 4, you infiltrate the plants. We grew 3 engineered end-to-end mammalian plants. We put our transformed bacteria into them, and the bacteria essentially transfer our gene into the plant. I can get into that more later if you want. We regularly water them. It was allowed to grow 22 degrees for 5 days. Had 16 hours of light, 8 hours of dark. The reason I specify that is that plants have light and dark reactions that they need to do to be able to survive and like grow healthily. That's why we're having it with light and dark. I believe Andy determined this experimentally. What was the no?
No, it's no.
Okay, well it works well. 16 hours of light and 8 hours of dark works pretty well for our purposes in our lab. If there's different data, maybe from the Moore lab, I'm sure we'd be happy to hear. So then, yeah, this is pretty cool. This is how you actually get the bacteria in. That's going to give the plant the gene. So first you prick the leaf of the plant, and then you take a syringe full of agrobacterium with a plasmid in it, and you can shoot the plant full of like this solution. It actually just goes into the leaf. Like the leaf actually gets a little bit, tiny bit bigger. I'm sure if you measured it. It comes out kind of transparent. So you got this fluid just filling this leaf up, and so this agrobacterium infects these leaves. Or it doesn't really infect, but it transfers the gene into these leaves. The plant cells make our protein. It's great. It's delicious. Here's how delicious it is. It's like wasabi. So we took 90 grams of leaf tissue. We blended it into RSC extraction buffer. That contains 100 millimolar tris, 50 millimolar salt, essentially, 10 millimolar ETA, it's a chelator divalent cations, I believe, which are used for protease activity. So we don't want that to happen. So we put those ETA in there to sequester all the ions. PMSF is a protein inhibitor. Triton is a detergent to kind of help us bust up the cell membranes. And we said that at a pretty basic, or not pretty, but fairly basic pH, of 8.0. And yeah, so we actually, this is what's left over if you decide to blend 90 grams of plant material at your home for some reason, and you filter it with cheesecloth, you get this much like just fiber and stuff, and this much soup. And that's the leaf. You can see that it's not really being decolored too much. There's a little bit of yellowing, but really not too much. It's a pretty healthy plant, as you can see. We're not, plant rights people can be okay. We're not hurting too many plants. So then we add phosphoric acid to a synthetic solution, and what that does is essentially it makes an acidic environment that causes some of the residues of the proteins in the plant to get exposed and kind of like clumped together out of it. Because we don't want like plant proteins, we want our proteins. And so you can see we're adding this protein, or we're adding this phosphoric acid, we centrifuge it down, and this is all the junk that's like left over. This is all the stuff you don't want to put into someone. It's probably not going to hurt them too much. You would hurt if you put phosphoric acid in, which is why we put the base back up, the intrinsics. But this is all the junk that we don't need. So all that's left over in that nice green soup is our, well, some plant proteins, obviously you can see it's still green, but mostly our protein after we run it through a protein G column. Protein G is a protein that can bind antibodies essentially. I don't know what specifically it binds to in the antibody. I'm pretty sure it's probably the Fc gamma receptor or Fc gamma region just because it's what everything binds to, and it's pretty conserved. And I think we get protein G from a virus or some kind of pathogen, and so it makes sense that it binds a constant region of the antibody. So these are the results. Did we do it? Yes, we did. We did spectrophotometry to analyze each sample, and we ended up getting a few elutions of over 1 milligram per milliliter, which is a fantastic concentration. A milligram of antibody will run you a lot of money. And this is very good. These are very good concentrations. This made me very happy. Andy and I were celebrating quite a bit. As you can see, it does peak at elution 3, but it doesn't really go down as much. So further elutions would probably be helpful. We could also, maybe raise the pH of the solution that we put into it. I believe if you have a higher pH, you might get better binding maybe. But I mean, going down from to 750, it's higher than elution 1. There's probably still some stuff left in that protein B column. So if we were to do this again, we would definitely, run through further elutions and also maybe make it a little more basic.
Brandon, how did you determine the concentration?
How do I interpret the concentration?
How did you measure the concentration? How did you?
We did a spectrophotometry of the
of what?
of
With
the RSE.
What wavelength?
We use an A280.
So this is measuring total protein, right?
Yes.
So how do you know that this is your very own protein?
That's an excellent question, and that'll be on the next slide. We did Western blotting and Tomasi staining to confirm that this is mostly the protein. And I say mostly because it's not 100% pure. We haven't done a silver stain to confirm that it's just the protein. But as you can see here, you can definitely see some smaller bands in the reducing conditions. Maybe not with the slow reds, but they're